about gene ontology, gene enrichment analysis. So let's begin with that. So for that you have to click on this website which is called ShinyGO, and the version is V0741. It is from South Dakota State University. So it is completely free to use and you don't have to log into this website. So let's begin with that. So here you can see it is the interface, and from here you can literally analysis everything like enrichment analysis, chart, tree. Also you can generate your own string and KGG pathways, etc. For that, this is the place where you have to put all your genes from your data sets, and also you can also analyze your RNAseq data, but for that you have to go to another website called IDEP. We will discuss about this later. So let's just begin with this ShinyGO website. So we'll be starting with GO enrichment analysis. So for that we need some genes. So here are, you can see here are some gene list. So this is my from my own experiment, and let me just copy all the genes. So Control C, and you come to this website and just paste here the genes. But remember, whatever the species you are choosing, you have to alter it from here. So these are the genes from Arabidopsis. So I will be choosing Arabidopsis as a plant here, as a model organism here. So the genes I have taken, it is from my own experiment, and then this genes belongs to the plant Arabidopsis thaliana. So I have to choose here the same thing. This is Arabidopsis thaliana. Now you just click the submit button. Here you can see all the genes and the enrichment, fold change requirement, and genes, pathway genes, all will be here. So you can see that there are almost 35 genes which belongs to the endoplasmic reticulum, unfolded protein response, also 239 genes which is cellular response to hypoxia. And for example, there are 3 pathway genes which belongs to L-lysine catabolic process to acetyl CoA or acetyl coenzyme, and there similarly there are multiple genes which belongs to some of the pathways. So it is the enrichment analysis. Similarly, you can go and you can click here to get the chart. Here you can see this is all the fold enrichment, and this is the gene list, and these are the amount of genes which belongs to this particular pathways. So from here you can change the color, you can change the height, or you can change everything. So just for example, this is the chart type. As you can see, this is lollipop. So we have selected lollipop here. That's it. So we have selected lollipop here. That is why the symbol is kind of lollipop structure. So I can change it from here. Let's example dot plot. Just wait for some time and it will automatically got changed. As you can see, it is now changed into dot plot. Similarly, there are multiple types, just like bar plot. As you can see, the theme got changes again. For example, from here you can also change the color, like I am changing this color to orange, and you can see here it's got changed. And similarly from here I also can choose the other color like green, and it will got changed into green color. So this is the chart type of things, which also you can easily download this chart by right clicking on your mouse. Just click it and save images, and you can literally save it in any folder like downloads, and also you can rename it chart analysis. Just save it, and from here you can check. As you can see, this is clearly visible, which can be used for your publication. I have done it earlier. And now let's have a look on the tree. So you click on this, it will take some time and it will generate the data. In a similar method, you can download by right clicking on your mouse, and you can save the image from here. Now from this website we can also generate the network analysis, like or from where, in which pathway which gene is connected and which gene is triggering which pathway. We can check it from the network. Just click on it and it will give you some network analysis. As you can see, these are some cluster of genes. These are some cluster of genes that responds to stress, protein phosphorylation, and some other categories which are not truly interconnected, but it will give you a general idea from where and from which pathway the genes in involved, the protein is involved. Similarly, for now, if we want to check the groups, I have to click on the groups, and here you have to choose again the geobiological process. Now it will give you the gene list with high level of geo category. So you can see these from my gene list. This website automatically sort all the genes, uh, and as you can see from my gene list, this website automatically sort out the genes and how the genes are response to some particular category. Like these genes all are giving responses to stress, these all genes giving cellular response to stimulus. Similarly, these are some all other categories you can see, and you can also plot this data, also download this data as an Excel file. Like you can from here, once you click on this download button, it will automatically convert it into Excel file and you can open it from here. So let's just have a look on this data. As you can see, the things got converted into an Excel file, and from this Excel file you can study which genes is triggering which response. And these all are the AGI codes. You can literally copy these things and paste into their website, which is the universal website to study about Aerodosis thaliana. So this is the thing. Now let's get back to the website again, and from here you also can generate plots. It will take some time again. Yeah, you can see this is the distribution of query genes on chromosome chi-square test P, and you can see the P value here. So again, this file you can download by right click on your mouse, and you can literally save the image for your publication.